Dr. Patra, you may start with the introductions.
Okay, thank you, sir. On behalf of the organizing committee, I welcome you all to our second session. Now I request Dr. Madhu TN, who is the Assistant Professor of Department of Veterinary Clinical Complex, Veterinary College, Bangalore. And also he is the participant of this training program. Now I request Dr. Madhu sir to introduce our revered resource person, Dr. Santra sir, who is from NCCS. So I request Dr. Madhu sir, please introduce our sir, resource person.
Who is going to introduce? Let me unmute them. Just a second. Dr. Madhu.
Yes, sir.
Just a second. You can unmute yourself now.
Yes, sir. A very good afternoon to everyone. It's my privilege to introduce today's speaker, Dr. Manas Santra, sir. So he's working as a scientist in the National Center for Cell Science in Pune. I would like to give a brief introduction about him. Dr. Manas Santra, he did his BSc degree in the year 1996 and MSc degree in the year 1998 from Vidyasagar University, West Bengal. And he also did his MTech in the year 2001 and PhD in the year 2006 from Indian Institute of Technology, Bombay. And from the year 2006 to 2010, he did his postdoctoral research from the University of Massachusetts Medical School, USA. And Dr. Manas Santra did his graduate research on characterization of folding and assembly dynamics of bacterial cell division protein under the guidance of Professor Dulal Panda, Department of Biosciences and Bioengineering. And from 2006 to 2010, he did his postdoctoral. He worked as a postdoctoral associate in the University of Massachusetts Medical School under the guidance of Professor Michael R. Green. And from the year 2010, he started working as a scientist in National Center for Cell Science, Pune, Maharashtra. And from 2010 to 2015, he worked as a scientist D. And from 2015 to 2020, he worked as a scientist E. And from 2021, he is working as a scientist F in the National Center for Cell Science, Pune, Maharashtra. So his field of research includes biochemistry, biophysics, molecular biology, genetics, cell biology, and epigenetics aspect of cancer pathogenesis. During his career, Dr. Manas Santra has obtained various honors and awards, which includes Ministry of Human Resource Development Award Fellowship to pursue his MTech from IIT Bombay, and he was awarded with a Junior Research Fellowship from CSIR to pursue his PhD from IIT Bombay, India, and he also obtained or awarded with IUBMB Sigma Aldrich Corporation Travel Scholarship Award for attending Forum for Young Scientists and IUBMB conference, which was held at Budapest, Hungary in the year June 2005. And he was awarded with the Best Research Scholar Award from the IIT Bombay for the year 2006. And in the 2006, he was also awarded with the Young Scientist Award in the New Biology from the Indian Science Congress Association. And in the year 2009, he was awarded with the Innovative Young Biologist Award for the year 2008 from the Department of Biotechnology, Government of India. And in the year 2010, he was awarded with the Ramalinga Swamy Fellowship Award from the Department of Biotechnology, Government of India. And again, in the year 2010, he was awarded with the Charles A. King Trust Award for pursuing his postdoctoral research fellowship from the USA. And in the year 2019, he was awarded with the National Bioscience Award for Career Development from the Department of Biotechnology in the Government of India. And in 2001, he was awarded with Fellow of National Academy of Sciences, Allahabad. So during his career, he has served as various life members of various societies, which includes life member for the Society of Biological Chemists, life member for Indian Association of Cancer Research, life member for Indian Society for Cell Biology, life member for Society of Indian Science Congress Association, life member of Indian Society of Translation Research, life member for Proteomic Society of India, life member of the Society for Free Radical Research India. So, sir has also obtained 2 patents. One is the US patent by the name of SRPX for the treatment of cancer, and Indian patent by the name of active methanic acid glycoconjugate compounds process for preparation and use thereof. In this esteemed career, sir has also published 79 international publications in various international journals, and also has published a book chapter by the name of Interplay of Ubiquitination and Simulation with miRNAs. So with this brief introduction, I welcome Dr. Manas Santra, sir, to deliver his seminar in today's afternoon.
So thank you, Dr. Madhu. Now we are really happy to have with us like a person like Dr. Santra, sir. Now, sir, we are eagerly waiting for your speech, sir. Thank you, sir.
Thank you, Professor Madhu, for nice introduction, and it is not really brief; it is huge. Okay. Also, I'd like to thank Professor Patro for actually arranging my talk. This talk. I'm working in this field last 15 years, the cell cycle and cyclins. In addition to that, we are working on genotoxic stresses, genome instability, cellular signaling. Okay, so this is one of my actually interested topics. It is in my heart. So today we'll talk about that cell cycle and cyclins. And please stop me wherever you have doubt or you have any questions. Just don't hesitate me to stop. Okay, because since we have time, we can we can discuss whatever point you have. So, as you know that all organisms, all living beings, originate from a single cell, and then multi-cellular organism they forms due to the division of the single cell. So, if we think about human life cycle, is the fertilized egg, single cell, then it will be fetus, baby, child, teenager, adult, and when stop dividing and other things will be in the old age, that time shrinking, everything is skin shrinking and hair, everything will be there. And ultimately when stop everything will be the death. And that is the fate of all the living organism. So as I said, the single cell will divide in the somatic division through a cycle, through a specific cycle. So this is schematic. The cell, single cell, if you think about it, it will synthesize, duplicate its genome, and then this duplicated genome equally divide, okay, and then forms the two daughter cells. So this happens through four specific phases. Okay, this is the first phase, is the growth phase or G1, or sometimes it is called gap phase. In this phase, cells will be prepared metabolically very active. All energy should be sufficient energy should be there for the cells, because the next stage, the synthesis phase, where the huge amount of checkpoint as well as the energy needed to duplicate the genome. Okay, this is called the synthesis phase or S phase. In the next phase, again, the growth phase, or you can say gap phase 2. And then in this phase, again, cells will find out, check all that things, the checkpoint are activated, all are fine. Okay, whatever the duplication happens in that synthesis phase, all are duplicated properly or not. Okay, if it is not, then re-replication will happen in this phase so that genome is properly duplicated. And then that it will come to that mitosis. And again, it is subdivided into few phases, the prophase, prometaphase, anaphase, telophase, and ultimately cytokinesis. So growth phase 1 or gap phase 1, synthesis phase, and G2 phase, the gap phase 2. This is known as the interphase of the cell cycle. Okay, so if we how we'll know the cells are in the which phase of the cell cycle? So basically here it is the immunofluorescence techniques is used to understand what the cells are which phases. So basically the pattern here, is the DNA, the genome is basically stained with the DAPI, which is the intercalating agent, DNA intercalating agent. So the pattern of the DNA DAPI staining will tell us the cells are in which phase of the cell cycle. So particularly G1 to G2, you can easily understand the cells are which phase of the cell cycle. And this DAPI is the green. And EDU, you know that this is the analog of thymine. So when cells will be in the synthesis phase, so new genome will be forming. So that time, it will, instead of thymine, it will be incorporated. So then the amount of incorporation, based on the amount of incorporation, you can know that which is the in the S phase, in the is it early, mid, or late phase. Okay, so when they are co-localized, that means the maximum co-localization is happening in the mid-S phase, and then again it will be down, and then the G2 phase you will not see any EDU staining anymore. And so same thing here, it is shown here. It's through another protein. It is the PCNA, proliferating cell antigen, nuclear antigen. So you see the pattern of the staining localization of this PCNA is distinct from distinct phases. You see that there is a speckle is forming, but it is a huge. But the much more foci formation, it will happen in the earliest phase. Okay, earliest phase, and then you see that there in the mid-S phase, their level will be little less, and it will be reduced again. And the latest phase will be the very few dots, and again in the G2 phase it will be smear. Okay, but in the G2 and G1, there is a huge difference. That here the spots are there, bright spots are there. I can't see whatever. Is there any question? Is there any questions happening, popping up? Okay, so I request that if there is a question, just because I in the presentation mode, I can't see anything. Okay, Professor.
No, sir, there are no questions right now. You can
Okay.
carry on. If there are any questions, they'll be in chat, and we can handle them at the end of the session.
Okay, because I can't see in the presentation.
Yeah, I know. I understand that. So we are able to see you. You are not able to see us. Yes.
Yes. So then in the mitosis, again, with the help of simple DAPI staining, we can know that which phase of the cell cycle. If it is the prophase, you see there is a nice arrangement of that our genome. And the pro-metaphase, it will be much more congressed, and then it will be a pattern. And pro-metaphase, if you have the confocal microscopy, you see that this kind of, if you the regular microscopy, you will see the mitotic plate that they are congressed. One plate will be there like that, but in the confocal microscopy you will see the nice arrangement, and it is outside like that, because the kinetochore, that microtubule is coming in the slide. So kinetochore will be there, and the microtubule will be attached. Okay, and in the anaphase, that equally that our genome will be divided, and then they will be segregated in towards the tupole. Okay, and this is the distance will be more in the telophase, and ultimately two cells will be there. So here, whatever I talked about, that in the mitosis, what kind of events are happening is the pictorial is shown here. Basically, prophase, you will see the condensation of the chromosome. Okay, it will be there, and the centrosome, it will be splitted, and it will be moving towards the pole. Okay, and all microtubule originating center, the gamma tubule and the microtubule will be originating. And then in the pro-metaphase, again, their distance between two centers will be increased. They will be almost placed towards the pole. And then these microtubule will start finding the kinetochore in the chromosome. Okay. So in the metaphase, again, here, this phase, the mitotic checkpoint will be activated until unless all the kinetochores is attached by the microtubule, and both side microtubule, the strength are not equal. The metaphase halt will be there, mitotic checkpoint will not allow the cells to proceed further to the anaphase. So they are huge, the checkpoint, mitotic checkpoint or spindle assembly checkpoint, both will be activated. And they will measure the both side the microtubule strength. Okay, and if it is not equal strength, it will anaphase will not initiate, because if the strength of the microtubule will be different, then there will be chance of aneuploidy of the cells. And once this is satisfied, then mitotic checkpoint will be inactivated, sac will also be dissolved, and then the early anaphase will start, and then at the end, telophase will happen and cytokinesis happens. So now if we think about the lower eukaryotes versus the mammals, these phases are almost, the 4 phases will be there, but depending on the complexity of the organism, the new factors will be involved in regulating the different phase of the cell cycle. For instance, the yeast, you see that one the cyclin dependent kinase, I'll come that in the few slides later. The one cyclin dependent kinase will regulate all phases of the cell cycle, and only 2 different cyclins are involved. Whereas in the mammals, in the classical model, there are 4 cyclin dependent kinase, the CDK. CDK is cyclin dependent kinase. So the 4 kinase, CDK1, CDK2, CDK4, and CDK6. And then 4 different cyclins are involved in the phases. And here in that mammals, that particularly the embryonic stage, the embryonic development stage, only one CDK is important, only the cyclin CDK1. And here the 4 cyclins are there. Okay. In the specialized cell, means like specific organ, also particularly here it is showing that in the CDK1 specialized cells, it will be CDK4 when it is the pancreatic beta cells. Like the specific cases, specific incidences, specific CDK will be involved. Okay, and if we disturb these, any of these, okay, there will be genomic instability, there will be chromosomal instability, there will be unstable proliferation, and that ultimately leads to the cancer. And as you know, that cancer is nothing but deregulated cell cycle progressions. So deregulated cell cycle progressions is the major cause, or is the I'll say phenotype for the cancer. Now, what are the, in the mammals, what are the molecular players involved in the cell cycle progressions? So the 3 groups of proteins are involved. One is the cyclin. Why it is cyclin? The name cyclin because the cyclic way they appear and disappear. Okay. So that's the reason that it is a cyclin that 1991 first time from the Harvard group, they showed that there is a proteins, Mark Krishner group, proteins which with the depending on the phase of the cells, so they are cyclic way coming and disappearing. Okay. So so this is the one, the cyclin. The other protein is the cyclin dependent kinase, the CDK. And the third category is that cyclin dependent kinase inhibitors. So they, depending on their things, either they will be binding to the cyclin dependent kinase or cyclin dependent kinase and cyclin complex, okay, depending on their nature, and that way they will regulate the activity of these kinases. But one important thing is that most of the kinase does not need to show their kinase activity through binding with the other protein. But cyclin dependent kinase is the unique until unless they bind to their corresponding cognate cyclin partner, they will not have kinase activity. That's the reason this way telling the cyclin dependent kinase. Without cyclin, they will not have any kinase activity. Okay. Then GM progression basically that controlled by cyclin dependent kinase 4, kinase 6, and they will bind to the cyclin D. And once they form the complex, they phosphorylate one of the protein, the first tumor suppressor protein, retinoblastoma. So retinoblastoma protein will be phosphorylated, and phosphorylation will inactivate retinoblastoma protein, and that will allow the protein like E2F transcription factors. There are multiple E2F transcription factors. Then those transcription factor will actually the regulates the proteins, even cyclins, the involved in the other phase of the cell cycle. And this cyclin D, CDK4, CDK6, this complex will work in that initiate the G1 phase to the late G1 phase. And then in the late G1 phase, the another complex, CDK2 and cyclin E complex will form, and that complex will actually helps the late G1 to S phase transition. And once cells enter into the S phase, the cyclin A and CDK2 complex will monitor the progression of S phase, because here that the source of genomic instability, the S phase is the source of genomic instability. So it will be precisely regulated in this phase, and then once it is the replication is completed, then G2 phase will be there. Here that the same cyclin will be there, but cyclin dependent kinase partner will be changed. It will be the CDK1, and then M phase, the early M phase will be again regulated by cyclin A CDK1 till prophase, and then after prophase, this will be the CDK1 and cyclin B. So any deregulation happens here, anything a problem happens here, this is the CDK cyclin inhibitor, the cyclin dependent kinase inhibitor, they will bind this complex, prevents their kinase activity, and thereby prevents the phosphorylation of retinoblastoma protein. Then retinoblastoma protein will be hypophosphorylated, and that will prevent E2F to prevent the E2F functions, that is transcriptional activity of E2F. So the genes which are involved in S phase and other phases, those will not be activated. Similarly, major other checkpoint will happen here in the M phase, that is the spindle assembly checkpoint, mitotic checkpoint, all will be activated here. If there is some abnormality, any abnormality cells will find, then this process will not happen, because this is the process where genome will be mutated. Here it is the genome will be destabilized because of the aneuploidy happen with the mitosis. So that's why, so far what I have spoke to you is basically the players, the G1 phase, the cyclin D1. In the G1 phase, there is three cyclin D1, D, the D1, D2, D3, and D3 specific organs it will express, but D1 and D2 are ubiquitous. Similarly, cyclin E, E1 and E2 is there, will come in the coming slide. So cyclin D forms the complex with CDK4 and CDK6, and cyclin E forms the CDK complex with CDK2, and these are the checkpoint inhibitor. In the S phase, basically early S phase, it will be regulated by CDK2 cyclin E, and then the major part will be regulated by cyclin A and CDK2 complex. Then in the mitosis, it is the, as I said, in the prophase, until prophase, the cyclin A and CDK1 will be the important kinase that we monitoring. And at the end of the prophase, the cyclin A will be inactivated and cyclin B will be activated, and this cyclin B will form complex with the CDK1, and that will monitor the progression of M phase of the cell cycle. So if in the mammals, we have almost 20 CDK, almost 20, but except 5, 6, the functions of other CDKs is not known. The functions are not well understood in that other CDK proteins, and what it is, so that's why the major focus about the CDK is the limited to these four and things, the few CDK5 also fine, but CDK5 majorly involves not in the cell cycle, mostly in that cancer. So CDK1, when it was knockout, that actually embryo, the 2-cell stage, it stopped growing. Okay, so it is embryonically lethal, though other CDK2, CDK4, and CDK6 are present. Okay. And that's the reason that you see the embryonic cell cycle, it is only depends on CDK1. It does not need other cyclin dependent kinase. In this, if CDK1 is there, but other CDKs is actually deleted, then also the embryonically lethal. Okay, so the development stage is done, but the other CDK is also important. So that's the we'll come to this. The latest slide also will show you the what CDK will be compensated by other CDK. So those that the CDK1 and CDK4 is there, other two is not there, but those animal, though they are normal, but they will be sterile. So here it is showing that the CDKs, as I mentioned, the CDKs kinase activity will not be there unless they form the complex with the cyclins, the corresponding cyclins. Here it is showing that the CDKs, what are the CDKs forming complex, what are the cyclins? Like here CDK1 can form the complex with the A1, A2, B1, B2. Okay, CDK2, it is many that it can interact with the A as well as the E. We have this. Also, specifically, it can interact B. CDK4 only cyclin D. CDK6 only cyclin D. CDK3, CDK5. Interestingly, the CDK5 so far, we don't know what the cyclins it interacts, and this is only Q11, and we have in our genome almost 20 CDK proteins. Okay, and as I said, that who can compensate which kinase it's showing here. Basically, arrow is showing is that this deletion in the G0 G1 phase. If CDK4 is depleted, this homozygous deletion happens, then CDK2, CDK6 can compensate and allow the cells to progress. In the S phase and G1S phase, if CDK2, again CDK1 and CDK4 and so on, but CDK1, if we deplete, then none of the other CDKs can compensate it. That means CDK is absolutely essential. There is no compensation. CDK can compensate others, but others cannot compensate CDK's function. So now, as I said, the main driver for the cell cycle is the cyclin because their level, as I said, the cyclin name came because the cyclic way of their appearing and disappearing during the cell cycle progression. And CDK's level almost remain constant throughout the all cell cycle phases. Okay, so their level, their only change happens. I'll come that the functions that happens in that because of post-translation modification. Okay. Post-translation modification, because proteins undergoes different types of post-translation modification, and CDKs generally undergo the phosphorylation. Phosphorylation is the one type of the post-translation modification. In my talk, we'll talk about the other modification also in the coming slide. This is the cyclins, basically, their activation mostly happens at the transcriptional level. And once their functions are there, they will be inactivated. And I'll show you the one data that if their stability is not precisely regulated, it induces the genomic instability in the cells. That any cyclins you can talk about, if their level is not precisely regulated throughout the cell cycle, then definitely genome instability will be there. So I'll show you a few research data regarding this. So if anyone want to halt the cells at particular phase of the cell cycle, so you can do that by down-regulating, suppose the down-regulating of the corresponding cycle. For example, if I want the cells to be arrested at the G1 phase of the cell cycle, then if I downregulate cyclin by any means, that then cells will be arrested, okay, and at the G1 phase of the cell cycle. And if you arrest the cells at the G1 phase of the cell cycle for longer time, more than 72 hours or so, then cells will understand there will be a serious problem in that. Then cells will be basically undergoing senescence, one kind of cell death, and cells will be lyse. Okay, and because of these, their properties, the cell cycle property, the most of the cyclin are known as oncogene in cancer. But in particular, cyclin D and cyclin E, the amplification is there, multiple copies happen, huge expression level is there, cyclin D and cyclin E, and these are known oncogene. But unfortunately, there is no inhibitors established till date available as a drug, because there is a problem is that if you inhibit the cyclin D functions or any cyclin function, that normal cell cycle progression also will be affected. So huge cytotoxicity will be there. Other cells, normal cells, will be also defective. But still, that other protein like the microtubule, as you know, the any combination therapy for the cancer, the one compound, if it is the chemotherapy, combination therapy, chemotherapy is given, that definitely the microtubule, anti-microtubule drug will be there. One of the compound will be there for the combination therapy. Why it is so? Why we can, microtubule also is very essential for the mitosis to equal division of our genome. But how it is happening? It is shown that the microtubule properties in cancer cells are actually different than the normal cells. Their post-translation modification is different. Their dynamics property is different. And basically, these mitotic drugs are basically affect the dynamics, microtubule dynamics, not the protein stability. So that's the reason till date, one of the drug will be the microtubule targeted drug, but we cannot target cyclins for the therapy, though we know that the cyclins are one of the culprit for the initiation of cancer. Okay, so here what we I'm showing the compensation as like cyclin, cyclin dependent kinase. The cyclins also can be compensated by other cyclin, and this is the table is showing that, as I mentioned, the G1 phase or G0 phase, resting phase, it is regulated by the mostly the D-type cyclin in the majority of the cells. So the D1, D2, D3s are there. Okay. So if D1 you deplete, then D2 and D3 can compensate same as this. If D1, D2 deplete together, then E1, E2, because as you know, the E1 and E2 is responsible for the progression of late G1 phase. Okay. Similarly, D1, D3, same thing will happen. Okay. And if we deplete, okay, this is the one science paper is there, on cell paper also there, that they depleted 3 cyclins, D1, D2, D3, and mouse development is fine. Okay, so in that case, that the E1, E2, and cyclin A compensate these 3 cyclins, cyclin D's in absence. Similarly, the G1S, as you know, that there is 2E, E1, and E2. So if we block that E1, then E2 can compensate similarly. And if E1, E2 is not there, then D1, D2, or cyclin A can compensate. And cyclin A can be compensated only by E, not by others. B cannot compensate the cyclin A. Because why I'm saying B? Because CDK1 can form the complex for B as well as A. CDK1. Since A is not, A1, A2 is not there, B is available, B can be, but B cannot compensate. Only E can compensate when you inhibit the A2 or A2. Cyclin B, again, here surprisingly, in the absence of B, B1, B2 or A can compensate, B2, only B1 can compensate. And if you do B1, B2, the mitosis cannot progression happens. So none of the other cyclin can compensate both cyclin and cyclin B deletion. And here we are showing the development defect. Development, as we said, the B1, if you do, A2, if A1 you do, the mice will be normal, perfectly fine. But A2, if depleted, that before E6.5, means embryonically lethal. So that D1, D2, D3 also that E14.5, still it is knowing, but this result is controversial, because there is a paper they are showing that the completion of complete deletion of this, the mice will be fine. So as I said, there is a checkpoint. Because they function, this checkpoint is there, basically they are breaks in the cell cycle progression. They halt the cells, help the cells to resolve the issues what arises during the progressions. So they will give the sufficient time for the cells to resolve the issue during the cell cycle progression. If, as I mentioned, if problems are persistent for a long time, then cell will decide and cell death process will be executed. So they are the breaker in the cell cycle progression. Okay. and they, depending on the nature of the cell cycle checkpoint inhibitor, they will either directly bind to that cyclin dependent kinase or cyclin dependent kinase inhibitor, cyclin dependent kinase and cyclin complex, to prevent basically the phosphorylation of retinoblastoma protein. As you can see here, that all the cases, retinoblastoma protein in the normal scenario is that it will interact with the E2F major transcription factor in the cell cycle progression. So it will form the complex, it sequesters the E2F activity only and only when cyclins and CDK complex phosphorylated retinoblastoma protein, then it will be pulse off. And once it is pulse off, then E2F with other regulated proteins, they will transcript the other genes. And it happens in almost all the cases, in the except mitosis. Mitosis, in the, as I said, there are two checkpoint protein complex are there, not a single protein. There's a complex, huge complex, the mitotic checkpoint complex and spindle assembly checkpoint complex. Even actually the other proteins also basically helps. But the one protein here, the multiple protein here written here, multiple protein can function as a checkpoint. In the M phase, only one protein, like cell cycle checkpoint inhibitor protein, the P21 major has major function here. Okay. Now, since we are talking about the cyclin and cell cycle proliferation, so the question is how to measure the cell cycle progressions. The three major experiments generally all cell cycle labs are doing is that first is the fluorescence activated cell sorting. This is the standard gold standard experiment. The three gold standard experiment. One of it is the fluorescence activated cell sorting. So here the propydium iodide used is the intercollecting agent. So it will be in that free form. It will not have any fluorescence. But when it will binds to the DNA, okay, so then that its fluorescence property will increase, and more binding, more DNA binding, more amount of, so more fluorescence will be there. Okay. So based on that, actually we can see the cell cycle analysis. There will be two major peak. This peak is the responsible for 2N chromosome, and when it is the synthesis happen, means DNA replication, so the cells deployed, that it will increase. So the cells in between this first peak and second peak, this is the S phase. Okay, and when genome is totally duplicated, then this phase is the G2M phase. Now actually, but quantification, this quantification, okay, again will not be able to tell how much cells are in the G2 phase and how much cells are the M phase of the cell cycle. So another alternative process is the BRD incorporation or EDU incorporation. If we do that, then the cells is a distinctly, okay. In the drawback in this is that you cannot isolate there, but here you can isolate the population. This is the G1 population, this is the S phase population, this is the G2M population. So you can separate the population here with this, particularly the EDU staining, because that the BRD incorporation. So, and these are basically the analog of thymine. So they will be incorporated. The easy is that the EDU staining, it is the less number of cells is sufficient to do the experiment. But BRD, you need huge number of cells, particularly that the cells like patient cells and all those, the BRD, the experiment will be difficult. It is small amount of cells, the EDU staining will be much better. So the another technique is that, again, it is not quantitative, it is the qualitative, it is the Western blot based method. So you know that cells, based on that, the which phase of the cell cycle, you probe with the marker like cyclin A. As we know, it will be from a cyclin, basically S phase to early prophase. So you know the cells, these the cells, the time point, you know, these are the cells in this phase. Similarly, the cyclin B, these are, and the phosphoserine histone 10, it will tell the mitotic phase cells. That measure the quantitative data you can get, mainly use the fluorescence based study. This is the two proteins, the kinase, the Aurora A and Aurora B kinase, which plays very important role in the progression of mitosis. But if you stain them, both of them, you'll see the pattern of their localization within the cells is distinct. So based on that, you can characterize the what are the cell. Okay. Now, as I said, the cyclins always fluctuate, they oscillate, they activate when the initiation of the phase of the where they are required, and then they are inactivated. So majorly the activation happens majorly due to the transcription level increase the transcription level, but their inactivation majorly happens through proteasomal degradation. What is proteasomal degradation? Basically, there are organelles called proteasome, 26S proteasome. I did not include those. So 26S proteasome, and in animal cells, human cells, that almost per cell there are 35,000 26S proteasome. It is kind of dustbin. It is kind of dustbin where all inactivated proteins or deregulated, misfolded proteins, all will be there, and there will be chopped. What we are now, our waste management systems are there, exactly within the cells, the proteasome systems is there. They will actually chop the inactivated protein, and all cyclins, or I'll say the 80% of our cellular proteins are inactivated through 26S proteasome. And it needs specifically how they will know this protein need to be inactivated. Basically, that is clearly demarcated by another type of post-translation modification. It's known as ubiquitination. Okay, so we'll come in the coming slide that how ubiquitination is happening. Okay, so if we think about like human cells, our life cycle, similarly, protein has also life cycle. They also born, brought up, then they do the function, they enjoy their cellular role. Okay, they enjoy. Okay, and then ultimately they have to die. Okay, similarly, or proteins will die. So most of the 80%, this is the proteasome schematically showing that when that endoplasmic reticulum, it is born and folded, then they will interact, they will undergo post-translational modification, interacts with the other protein, so they're grown, and then those will be the functional. And then once they function, then when they need to die, they will be ubiquitinated. Okay, and then those proteins will come to the 26S proteasome, it will be chopped here. Okay. Otherwise, it's the special case like nutrient deprived case or other endoplasmic reticulum ER stress. That time lysosome plays major role to inactivate the proteins. So if we think about it, the protein, okay, like us, protein has equally similar pattern of life. So once it is synthesized, chaperone is there, they will bring folded. This is this stage I can say this is the fetus. Okay, and then once it is folded properly, this is the baby, a protein folded, and then once it is folded, several types of post-translation modifications are happening. Lipidation, sumoylation, phosphorylation, acetylation, so and so forth. The one thing for the demarcation, for the death, the ubiquitination must be there. But different types of ubiquitination happens. So one type of ubiquitination will mark this protein to be for the death, degradation. Like once it is folded, that post modification happens, then teenager is telling that basically protein interaction, they are friends like that. Okay, then they will interact, the adult stage, they will interact and do many things within the cells. And then once they're done, their functions is done, now they are redundant for the cells, they are burdened for the cells. So then will be ubiquitinated, okay, and then this will be degraded. The protein death will be there, okay. So as I said, the 26S proteasome. This is the each cell, mammalian cell. Each cell will have basically 35,000 26S proteasome. It has the regulatory subunit, both sides, and it is the catalytic core. Here, all the ubiquitinated proteins, suppose this is the ubiquitinated protein, it will enter there. Then here the enzyme will be there, few enzyme will be there, they will be deubiquitinated. And then this protein will be transported in the central part. And where all proteases are there, this will be chopped and it will come to the amino acid. And this ubiquitin will be recycled. So this is the ubiquitin, very small protein. This is the two types of modification where protein is involved. The ubiquitin is one and simulated also is the one. These are the small protein which will be covalently conjugated to the protein. So in 1975, the first discovery of ubiquitin was in 1975. And you know, the Goldstein actually is the first author of this paper, and he is the also corresponding author. So he, when actually isolated different organelles, and then he resolved the protein fraction whose molecular weight is from 4,000 Dalton to 12,000 Dalton. And then they found that the one protein which is the ubiquitous for all the tissues, that is then their group basically named that the ubiquitous immunopoietic polypeptide. Okay, this is the ubiquitin protein. Okay, later on it is crystallized, and and then this protein, the ubiquitin, this is the, as I said, 76 amino acids are there, and 6 lysine acid residues are there. Position 6, 11, this 3, and this 2, T48 and lysine 63. So these these lysine residue, ubiquitin used to form the ubiquitin polymer chain. Okay, like that here, lysine 63. So proteins may be monoubiquitinated, proteins may be multi-monoubiquitinated. This is a multi-monoubiquitination, but all ubiquitin is directly attached to the proteins. Here, the polyubiquitination is happening. Only one ubiquitin is basically, I don't know. Okay, so basically here the protein is getting polyubiquitinated. That only one ubiquitin is directly interact with the substrate protein, and the other ubiquitin is conjugated to the first ubiquitin. So there is a polymer formation happens, and when this polymer happens through lysine 48, then this protein will be marked for proteasomal degradation. Same thing from the lysine 11. The lysine 48 basically it's prevents the degradation. One. Second, it's also the if proteins are undergoing lysine 63 based polyubiquitination, their cellular localization change and huge the repair process, stress response process also involved. Okay, and that we don't know the functions of others, but our lab is working in this, that we are trying to understand the importance of this conjugation. So these are the three scientists, okay, who got Nobel Prize because of their work on the how ubiquitination is happening, what kind of structure and all those things. So though ubiquitin discovery was done by Goldstein, but the functional characterization, everything is done by these three scientists. So they got the Nobel Prize. Okay, as I said, this is the ubiquitination is distinct from the other post-transfer modification. How? Because here in that, for example, the post-transfer modification, phosphorylation, protein phosphorylation, that is basically substrate protein will interact with the kinase. Kinase will interact with the substrate protein, and then phosphate group will be transformed from ATP. But here it is three cascade enzymes are there. Okay, three-step process here. The first, ubiquitin will be activated in the presence of ATP by E1 activating enzyme, and we have only two E1 activating enzyme. Then this activated ubiquitin will be transferred to E2 ubiquitin enzyme, and we have almost 40 E2, but till date we don't know the functions of 90% of E2. Okay, and then this activated ubiquitin will transform from E2 to the substrate protein, which need to be ubiquitinated. Here it is showing polyubiquitination with the help of another type of ubiquitin enzyme that is called E3 ubiquitin ligase complex. It is mostly multi-protein complex happens, also single protein complex happens. And in the human genome, we have almost 600 E3 ubiquitin ligases, but majority function we don't know. This E3 basically recognize the substrate, bring the substrate, and then it will be ubiquitinated. And they are further classified into four classes: Heck type, ring finger, Ubox, and pH. And our cell cycle progression basically regulated by ring finger E3 ubiquitin ligase. And these are further classified into many groups, and majorly cell cycle progression is regulated by anaphase promoting complex and SCF complex. If these complexes are not there, cell cycle progression cannot happen, because the proteins, particularly cyclins, they will not be ubiquitinated. Once they will not be ubiquitinated, they will not be degraded. So the inactivation will not happen. So that's why the cell cycle progression, the cyclins level at the post-translation level regulated by APC and SCF complex. What are those? Basically, APCC complex, this is called anaphase promoting complex. This is the 14-member complex. Okay, 14-member complex, and if you most of them are very important. If you deplete them, the mouse will be embryonically lethal. Okay, so in this complex, the two proteins which recognize the substrate are the CDH1 and CDC20. They brings the substrate. The basically here, how they recognize the substrate? If the substrate has the specific motif called RXXL, R is arginine, and any protein, both protein, and then last L. So those proteins will be recognized by these either CDH1 and CDC20. They will bring to this, and then it will be ubiquitinated. Whereas SCF complex, this is the quaternary complex. The three members are conserved. Therefore, so here the SKP1, this is one protein, Kalin proteins, and then ring finger proteins, RBX. So these are the actually invariable component. The variable component basically Ebox protein. SCF because SKP1, Kalin 1, and this is the Ebox. Okay, I'll come in later slide. So they are basically recognizing the substrate. Okay, so if we think about now cell cycle, so 4 phases. The APCC CDH1 basically monitor the progression of G1 phase and late M phase. Whereas APCC CDC20, sorry, here most probably something happened. So APCC CDC20 that regulates the mitosis. So and all the other phases previously it was thought that, means 10 years back, it was thought that the APCC CDH1 is the only regulator of mitosis and G1 phase. But now we know that there are multiple complexes are involved the progression of G1, S, and G2 phase. So SCF complex actually, they regulate all the phases of the cell cycle, but G1, the APCC is restricted to mitosis and G1 phase of the cell cycle. So what is SCF complex? Sorry. So as I said, these are three proteins are conserved. They will be invariable. Only variable is the Ebox protein, and they functions as a substrate receptor in this complex. Majorly, sorry. So Ebox proteins majorly recognize the phosphorylated substrate. Once they recognize, they brings the substrate to the complex, and with the help of E2, ubiquitin will be transferred. And based on the protein, if it need to be degraded, then either K40 or K11 linked polyubiquitination happens, and then it will be actually sent to the 26S proteasome, and there it will be degraded. Now, that in the human genome, we have 69 Ebox protein, and that only 7 proteins so far we know the functions of detailed function of only 7 Ebox protein. We don't know the function of other Ebox protein. So they classified into 3 groups. One group is that ABXW, because they have WD repeat in their C-terminal domain, and mostly these domains are involved in the protein-protein interaction. Similarly, ABXL, leucine-rich repeat domain will be there. Again, leucine-rich repeat domain is also responsible for the protein-protein interaction. And when the Ebox proteins, they don't have the specific type of domain, they are called the ABXO or other domain. And these all the proteins will have the common motif called the Ebox motif. This is the 40 to 50 amino acid protein, and this is important for the interaction with SKP1. As I said in the previous slide, the Ebox protein will associate with the SKP1 through their Ebox domain. If Ebox domain is not there, then this SCF complex formation will not happen. Okay. So today I'll discuss only one protein functions that the cell cycle. In that, this is the ABXO31. What happened? I'm sorry, something is. Sorry, there is some problem. Just. Are this slide visible now?
Yes, sir. It is visible.
Okay, okay, thank you. Okay, so that FBXO31, what it was known before 2009, it was known that it is a member of Ebox protein family and it could function as a tumor suppressor, putative tumor suppressor, because if you express this protein, cells undergo senescence. The senescence is the one type of cell death. We know the cells undergo death by majorly by 4 processes. One is apoptosis, one is necrosis, one is autophagy, another is the senescence. Necrosis, where the immune response will be huge, whereas autophagy less, senescence there will not be, and apoptosis also, the immune response will be very less. Okay, the senescence, the distinction from other cell death mechanism is the senescence require cells to be arrested either at the G1 phase of the cell cycle or M phase of the cell cycle. So as I discussed, the arrest the cells at the G1 majorly will happen either down regulation of cyclin D1 or activation of cell cycle checkpoint inhibitors like P16, P14, P19, P21. Okay, so their activation. And in 2008, okay, it was actually shown that their paper, they showed that if BRAF oncogene is introduced in the primary cells, then if you deplete the FBXO31, then oncogenic transformation will increase a lot. And FBXO31, if it is there, it will prevent the oncogene to transform the primary cells into the transformed cells or malignant cells. It induces the senescence. So these two events, said that FBXO31 might have a role in the cell cycle progressions. So this telling that since the oncogene introduction, basically what will happen? Oncogene will facilitate the progression, and by doing that, what they do, they generally shorten the replication process. And during the sorting of the narrow down the S phase, so there will happen the replication pause, stalling will happen, and that will generate both single strand and double strand break. And then the DNA damage response protein will be activated and cells will be arrested either G1 phase or G2 phase of the cell cycle. Why that arrest is important? Because that arrest, the halting, will allow the cells to repair their genome, whatever damage has happened. So we thought that whether FBXO31 has any role in that cell cycle progression. So what we have done, simple experiment we have done. We depleted FBXO31 using the shRNA, a short hairpin RNA. So that will reduce the FBXO31 and then synchronize the cells and allow the cells to progress. And you see, there is certain level that progression is slow. In that G1, we arrested the cells G1 phase, and then M phase, you see the M phase is less, both when you depleted the FBXO31 in using two different shRNA. Why so? Then we check, since S phase progression is happening, okay, what is the reason? So we checked the cyclins, we find that cyclin level is increased. Okay, and if we except cyclin B, all cyclins are nuclear protein. Okay, so majorly they will be the nuclear. So we fractionated it and check that cyclin A, when you deplete FBXO31 using the short hairpin RNA, we see that there is an increased level of cyclin A. Okay, and then further prove that we measure the turnover, protein turnover, using the cyclohexamide assay. Basically, what will happen? Cyclohexamide will stop the new synthesis. So there will be no new protein. Okay, so whatever existing protein, they will slowly they will be degraded. So by measuring that, we found that when we deplete FBXO31, then the half-life, the turnover is slowed down. So half-life increases from almost double the half-life increase. So that's telling that FBXO31 basically regulating cyclin A at the post-translation level. Okay, if it is at mRNA level, transcript level, this kind of turnover will not change. So then we said that why it is so. We also check that is it also cell cycle dependent. So we see that when that the cells were nocodazole arrest. So nocodazole is that microtubule targeting agent. What it does, it depolymerizes microtubule. Okay, depolymerized microtubule. So then microtubule will be only tubulin. So then all the cells will be halted at the metaphase plate. Okay, because there will not be chromosome, kinetochore will not be attached by microtubule, so that the congestion will not happen. So we see that the cyclin A level is increased when you deplete FBXO. This is the quantification. You see that there is a huge increase almost all phase of the cell cycle. Then we said, what is the problem? Okay, why S phase? Is that replication defective? So replication defective, one protein complex, a mini chromosome complex protein, there are multiple protein in this complex. We monitor in that one protein MCM4, because this is the protein which actually plays essential role to actually origin, replication origin pairing. So then the origin initiation is this loading protein, loading is essential. So if we see that if we deplete FBXO31, loading of MCM is actually reduced as compared to the wild type cells. Non-silencing means that we express the is a short hairpin RNA which will not target any gene. So that's why it's a non-specific or non-silencing. Okay, so with respect to that, but when we co-depleted, as we know that depletion of FBXO31 is increased the level of cyclin A. So is it due to the cyclin A level, more level? So when we co-deplete, means in addition to FBXO31, we actually depleted cyclin A also, and then we see that basically resemble that the MCM4 complex, the loading is getting resolved now when we deplete that. So this is the chromatin fraction and this is the non-chromatin fraction. So the chromatin fraction, we see the simple loading is getting reversed. So that means the origin, recognition and firing is problem. So that's why the progression of S phase in FBXO31 depleted cells are defective. So this is the model proposed. What we did, the FBXO31 in the cell cycle, it monitors the cyclin A level by polyubiquitinating the cyclin A. I have not shown those data. Once it is there, then the MCM complex will be loading there is precisely, so the replication initiation will start. But if we deplete FBXO31, then the polyubiquitination of cyclin A will not happen, and that will interfere with that loading of MCM complex. And as a result, the replication will not be happening properly. And also, as I said, that it also affects the prophase also, and then incomplete replication also affect the M phase. So that's why we see there will be lagging chromosome. The chromosome alignment also defective. So that's the way. Not only activation, deactivation also cyclins is very important. This is the one example. So another, okay, I'll skip this. As I mentioned previously, that FBXO31 is involved in preventing the oncogene-induced transformation. If FBXO31 is there, if you introduce oncogene, all cells will be senescent. Majority of the cells, I'll say, because only less than 0.1% cells transform. Other cells will be dying by one of the mechanism, either apoptosis or senescence. So since the observation 2005 paper is there, and we know that senescence induction prerequisite for the cell cycle arrest. So we asked whether FBXO31 is involved in the DNA damage response. So first, to understand whether it is involved or not, we actually examine that FBXO31 level. So any kind of, these are the known cancer therapeutic drugs, and these are the radiation always we use for the cancer. So we think that whatever the drugs you give for the cancer treatment, so all the drugs will induce the FBXO31, the level is increased significantly. And all these DNA damage, there is accompanied by cyclin D1 degradation. So cyclin D1 degradation means there will be cell cycle arrest will be there at the G1 phase. So this is the different region, and this is the different time point we have checked. And then, okay, if, as I said, in the DNA damage pathway, basically the gamma radiation, it induced the double strand break. And then double strand break, this is the kinase, the ATM or ATR is the master regulatory kinase. They activate, and then more than 700 proteins will be phosphorylated within 15 minutes of the DNA damage. And this kinase basically regulate this phosphorylation. And depending on the DNA damage, either cell death will happen, okay, apoptosis, or cell cycle arrest will happen, and that arrest allow the DNA repair, and then DNA repair will happen, then cell will be survived, and cell will come back to the regular cell cycle progression. So then we asked that whether any kinase is involved in the DNA damage response, and then because FBXO31 is getting stabilized. So these are the kinase, as I said. The simple experiment we have done here, we have depleted ATM using shRNA and examined the FBXO31 level. So as you see that in the wild type cells, the FBXO31 stabilized, but when we depleted ATM, then the FBXO31 stability is not there. So that means ATM actually responsible for FBXO31 stabilization following DNA damage. And if FBXO31 is not getting stabilized, then cyclin D1 is not getting degraded. That means FBXO31 actually involved in the cyclin 1 degradation during DNA damage. As a result, cells are getting arrested at the G1 phase of the cell cycle. So how does, then we actually, I'll go, then we examine the protein sequence, whether any sites are there for ATM or ATR to phosphorylated FBXO31. We find two sites. The one is the 278 position and one is the 40 position, 404 position. So basically 400, these two sites are the ATM phosphorylation site. ATM or ATR generally phosphorylate the protein if there is a S or T, serine or threonine followed by glutamine. Okay, then ATM or ATR can phosphorylate. So we found basically the 278 position is getting phosphorylated by ATM. So this is the basically in vitro kinase we have performed. Okay, so far what we have shown that ATM is getting activated and FBXO31 is phosphorylated, and that phosphorylation stabilizing FBXO31. And we know this is the progress happening. Then we ask that does FBXO31 play any role in the cell survival following radiation? And very interesting result we found that when you deplete FBXO31, cells are very lethal. Cells are not forming colony. Okay. So that means cell death is happening. Cell death is happening. So I'll say, I'll go open. Then we say that if radiation is happening, okay, radiation happening, cell cycles should arrest. So what we said that then FBXO31, whether involved in the cell cycle arrest, as expected previously known also that if you induce the DNA damage, cells will be arrested at the G1 phase of the death. This is a very simple experiment we have done. What we have generated FBXO31 depleted cells. Then we have actually those depleted cells exposed to the gamma radiation which induced DNA damage, and then those cells treated with the nocodazole. So if we treat the nocodazole, then if cells are not arrested at the G1 phase, then those cells will be arrested at the G2 M phase of the cell cycle. So we see that if we deplete FBXO31, majority of the cells are arrested at the M phase of the cell cycle. Whereas in that wild type cells, there is a fraction of significant population of cells are the G1 phase of the cell cycle. That means FBXO31 plays important role in the G1 phase of the cell cycle arrest. Then in the 2000, there's a Netherlands group, Rivenagami group, they showed that the G1 cell cycle arrest is happening and P53 major role playing the maintenance. But the initiation of G1 phase is not P53 dependent because they have used that P53 knockout cells and wild type cells. So both P53 knockout and wild type cells, the G1 phase is increasing with time, but after 10 hours, the P53 minus cell line, they are the arrest is going down again. But if P53 is there, the arrest is increasing. And they found that whether P53 is not there or there, cyclin D1 degradation will happen. And we found that FBXO31, if it is not there, then cyclin D1 degradation is not happening. And so it's showing here that if FBXO31 is deplete, the cyclin D1 degradation will not happen. Okay, and this is done by a different cell line basically. sorry. So what we have shown that if you induce DNA damage, ATM will be activated, and that ATM will phosphorylate BXO31 at 278 position. And once it is activated, it will target the cyclin D1. Sorry, MDM2, I have not shown that because this is not cell cycle related. So the cyclin D1 will degrade, and that degradation will induce the P53 activation, and that P53 activation also induce the cell cycle arrest. And this is, I'll skip that because here what we are showing that it is the degraded polyubiquitination is happening. So what we have shown, basically what we understand, that FDXO31 is getting phosphorylated by ATM. At the same time, ATM also phosphorylated MDM2. I'm not so, I have not shown, and cyclin D1. Sorry, that model actually, and that leads to degradation of MDM2 and cyclin D1. The cyclin D1 itself also sufficient for the P53 minus cells, so that will arrest the cells at the G1 phase, and if cells are arrested at the G1 phase for longer time, then cells will go for the senescence. I'll skip now. Okay. I'll skip here because these are the DNA damage. Okay. I'll go to the actually summary slide. So what I have shown is that the FDXO31 controls the basal level of cyclin A. So if you deplete the FDXO31, then cyclin A level under normal condition is getting increased, and that will prevent the origin of replication initiation as well as the origin firing, replication origin firing, and that creates a replication stress and cells are lethal. Okay, so that's why the cyclin regulation, cyclin A regulation, also very important. And I've shown that the FDXO31 actually targets cyclin D1 under DNA damage condition, and that facilitates the degradation. Cyclin D1 degradation also helps the arrest the cells at the G1 phase, and that G1 phase arrest helps the cell to repair their genome. That part I have skipped because those are not cyclin related one. Okay, and then that skip basically it is also target the gamma is 2x. Okay, it basically prevents the accumulation of gamma is 2x under normal condition, so that unnecessary cell cycle checkpoint will not be activated. Okay, and unnecessary accumulation of repair proteins will not be there. So what we understand basically, if FDXO31 is there, it will protect the cells from genomic instability. Okay, basically this is the model. Okay, it targets under DNA damage condition cyclin D1, and under G1 cell cycle arrest and senescence happens. Under unstressed condition, it regulates cyclin A to maintain the genome stability, to actually precisely regulate the replication process. Okay, so this is my lab, and so majority of work I actually presented. This is the Srinath's work and the Parul's work and Rajesh's work. Okay, majorly I have presented today, and these are my collaborators. So the Devashish Mitra from NCCS, Sikant Rapole from NCCS, Santanu from IIT Bhubaneswar, Praveen Kumar from NITI University Mangalore, Rajesh Gatche from Pune University, then Devashish Manna from IIT Guwahati, Mayur Kalari from ISAR Pune, Michael Green, still I have collaboration, and Subal Manna from Vidyasagar University. And I got the funding from the National Center for Cell Science, Intramural Fund, Department of Biotechnology, Department of Science and Technology, and CSR. And thank you. I'll be happy if you have any doubt, because I don't know, is there any questions are there in the chat box.
Sir, actually there is one question, not question. This is from Susita Rajkumar. Written, I am Dr. Susita Rajkumar, scientist, veterinary pathology, ICAR, CCARI, Goa.
Okay.
So he asked, is confocal microscope facility available at your institute? Can researchers from other organizations can use this?
Yes, it is available. Actually, this is the payment basis. Okay. Because you know that new government rules, even our internal, we have to pay. Okay. So for academics, different charge, for the industry, different charge. Okay. So there is our confocal is there. It is available. I think 3 days in a week we have slot. Afternoon slot is there. So
Okay.
it's not a problem. It is accessible, and the Arun Kartik is that in charge. Okay, he is actually maintaining that this industry or academia, those things. And it is available, no problem.
Okay, sir. And there is another one suggestion, that is, please share the PowerPoint after the presentation. It is very much informative. So as per our the rules and regulation, there is YouTube. YouTube, it is available.
So what I'll do, that there are some part are unpublished work. So I'll delete those and then I'll send you.
Okay, sir.
Because those are unpublished. We have not communicated yet.
Okay, sir.
Okay, so that small person, but major is 90% part will be there. Only the this part will not be, the repair part will not be there. Okay, because we have not published yet.
So, and I request our participants, please do questions to our honorable resource person. Now it is open, unmute, or you may raise your hand. Okay, then we will.
Is it understandable? First of all, there is no question is two reason. Is there not understandable at all, or it is understandable?
There is no reply, sir, still. So tomorrow, so now this is the vote of thanks to our resource person, sir. So really, we are very much thankful to you, sir, for providing such a nice presentation. So I want to share that during our secondary and the higher secondary studies, we generally learned the different mitosis, meiosis, and etc. for our study during our study. And when we entered into the specialized subject, that is in our pathology course, there we learned the cell, their cell injury, as well as their inflammation, and thereafter the repair process. After the cell injury, some few cells undergo apoptosis and few cells become damaged and few cells become regenerated. So, sir, during the regeneration process or the multiplication process, so we learned G1, S, G2, and M phase, and thereafter the different implications of cyclin A, and which was first discovered by Goldster and Nobel Prize, etc. What, sir, you have told. But up to, sir, multiplication, we have learned, we already know, and this was the course syllabus in our UG as well as in the PG syllabus.
Okay.
So sir, thank you very much, sir, for providing a nice presentation, as well as you have shown the one model, that is the FBXO31, which is from its sequel up to the sequel of the cell and its implication, as well as the oncogenesis different steps. What you have given, that is really, we have learned a lot. Sir, thank you, sir, very much for giving nice time to us.
Thank you.
So another one information to all our participants, that tomorrow will start at 10 a.m. Please positively attend. Okay.
Okay, thank you. Thank you all.
Thank you, sir. So closing this program.